Hi, and good afternoon. My name is Arthur Demrich. I'm the director of the Lemelson Center for the Study of Invention and Innovation at the Smithsonian National Museum of American History. I'm delighted to welcome you all to the sixth in our 12-session series of Pandemic Perspectives. I want to give a special call out this afternoon to members from Smithsonian affiliate museums around the country, as well as to people from the Smithsonian Regents and board members who are attending today. This series overall is a chance for us to bring experts together to provide some historical perspective on the current pandemic, and by looking at some selected special events from the past to give a better understanding of what we're all experiencing in the present. It's also a chance for us to give some background tours of Smithsonian artifacts. So those of you missing the opportunity to go to museums get a chance for a quasi-museum visit. And third, it's a chance through the question and answers, which we encourage you to submit questions through Q&A, to get a feel for some of the issues people are facing around the country, and for us as museum professionals to begin to think about what it is we ought to collect in the future. So this is a chance for us to have a kind of virtual dialogue about the present, to think about what we should be gathering for the future. Today, I'm especially delighted to introduce Keith Wayloo. He's the Henry Putnam University Professor of History and Public Affairs at Princeton University, and will be moderating today's session. His research straddles history and health policy with work on pharmaceuticals and drug policy, the politics of race and health, the interplay of identity, ethnicity, and gender with medicine, and controversies on genetics and society. He has authored or edited 10 books and 20 articles, along with numerous commentaries and essays. Among other work in the profession, he's the current president of the American Association for the History of Medicine. He holds a PhD from the Department of History and Sociology of Science at the University of Pennsylvania, where I in fact met him when I was a young undergrad. He's part of the reason I'm here today and got inspired to study in this field. And he holds a BA in chemical engineering from Yale. Keith, welcome, and over to you.
Thank you, Arthur, for the introduction, and I look forward to what promises to be a fascinating conversation. Let me introduce our panelists very quickly. First, we have John Grabenstein, who is Colonel John Grabenstein, is a pharmacist and vaccinologist, public health leader, and founder of Vaccine Dynamics. During a career that spanned Army service and directing the Department of Defense's global vaccination programs, and then leading global medical affairs for Merck Vaccines, and now as founder of the consulting service Vaccine Dynamics, John published over 300 articles and 11 books. He continues to edit weekly newsletters for the Immunization Action Coalition. Grabenstein wrote the curriculum for pharmacy-based immunization delivery, which is a CDC recognized course that has trained over 360,000 pharmacists. We also have with us Paul Offit. Dr. Paul Offit is the director of the Vaccine Education Center at the Children's Hospital of Philadelphia, as well as the Maurice R. Hilleman Professor of Vaccinology and a professor of pediatrics at the Perelman School of Medicine at the University of Pennsylvania. Dr. Offit has published more than 160 papers in medical and scientific journals in the areas of rotavirus-specific immune responses and vaccine safety. He is also the co-inventor of the rotavirus vaccine RotaTeq. In addition, he is author of 9 medical narratives, including Vaccinated, One Man's Quest to Defeat the World's Deadliest Diseases, and Deadly Choices, How the Anti-Vaccine Movement Threatens Us All. And finally, we have with us Diane Wendt, who is a curator in the Division of Medicine and Science at the National Museum of American History. She specializes in the history of pharmaceuticals and related health products in the United States. She has curated exhibitions on patent medicines, Civil War nursing, biotechnology, scientific glassware, and medicine during the First World War. Recently, she led an effort to create a web-based catalog of the museum's collection of historic vaccines, immunotherapies, and diagnostics related to infectious disease. And so with that, I would like to launch us into a conversation on the history of immunology, history of inoculation, and the history of the thing that we now call vaccination, with this event from roughly 300 years ago. That is to say, 300 years ago this year, in May of 1721. One might call it the first instance of both inoculation in the early colonies, but also how practices of inoculation come in a wide variety of forms and have often been contentious. And so this is an example, and let me just say a little bit of it, is of the time when the famous or infamous Boston cleric Cotton Mather learned from one of his slaves, a man named Onesimus, that based on having seen a scar on his arm, of the fact of the use of variolation, the use of smallpox pus moved from one sick person into the arm through a wound in a healthy person, to practice an early form of variolation or inoculation. It was both famous and infamous as the first kind of, clinical widespread use of this vaccine or this early inoculation variolation practice. And it was also a kind of an early clinical trial around which there was much opposition. And the images that you see here are the pamphlets, both arguing for and against this practice. Cotton Mather got a physician in Boston, Zabdiel Boylston, to inoculate 242 people, 6 of whom died, a 2% mortality rate, which is compared to the 14% mortality rate from the cases of smallpox incurred naturally. I wondered if any of our panelists, perhaps Paul or John, want to talk about this famous and / infamous event in the nation's history as a launching point for our discussion of the history of what became later known as vaccination.
I'll start, Keith, and just comment that to learn from the enslaved man is a clear reflection of needing to always keep your eyes and ears open, and you never know where you're going to learn something useful. So thank God that he did.
Mm-hmm. There's also an interesting story of how heated the reaction was to this practice. Someone threw an incendiary device through the window of Cotton Mather, which did not explode, and carried the letter that said, "Cotton Mather, you dog, damn you, I'll inoculate you with this, with a pox to you." Which highlights the fact that these practices, variolation, inoculation, and later vaccination, have always stirred public passions in an extraordinary way. And as John mentioned, learning from exotic practices around the world is also part of the origin point of these. So let's flip now to the kind of the long history of smallpox, which 60 years later, an English physician named Edward Jenner discovers that milkmaids, or so the story goes, milkmaids who had been ill with cowpox appeared to be immune from smallpox, and the use of cowpox in vaccination became widespread, particularly starting in the 1790s, more widespread, although it was discovered in the 1780s, and also subject to caricatures like this. John, observations?
So this is what the internet might have looked like in 1802. It's a political satire cartoon, as you pull out. Jenner is standing there just left of center, preparing to vaccinate the poor. And so the first slide we talked about just before was some of the vaccination being against God's will being the claim. Here, this is more of a safety question of you're going to have cow parts grow out of me. As you can see, the cows coming out of people's mouths and various body parts. But it's also a satire or a criticism of using government funds to give medical treatment to the poor instead of them figuring out how to pay for it themselves. So there's all sorts of social overlay that appear here.
The only thing I would add, John, is that we look at a cartoon like this and we find it ridiculous, right? That people would, after being inoculated with cowpox, start to take on physical characteristics similar to cows. But I would argue that much of what we claim today is about as biologically plausible as this. I mean, the notion that vaccines cause autism or diabetes or multiple sclerosis, or that the SARS-CoV-2 vaccine is going to cause infertility, et cetera. I mean, it's about as biologically sound as this, or that the mRNA vaccines will alter your DNA. It's all, this is it. I mean, I think people are just frightened whenever you inject them with a biological, and so their imaginations run wild.
Yeah, and speaking, sorry, go ahead.
I was just going to point out a few of the details in the cartoon too for people to focus in on. And one is in Jenner's hand, the vaccinating device there. It's actually a lancet that he's using. And also some of the devices that we see on the table on the left-hand side, the enema syringe, the tub of opening mixture, and a bottle named labeled vomit. Just to mention some of the other sort of very common medical procedures of the day. And the vaccination, before one was vaccinated at this time, the body was prepared through these sort of cathartics and emetics. And so just some interesting devices and things that also show up in this cartoon.
Great point. And Arthur pointed out to me earlier one other feature, which is the wall. There is hanging a painting of people praying to the golden calf. And this mixes, of course, both the religious and the cowpox and the scientific imagery together as a part of the critique as well, a kind of sophisticated symbolization of the critique. So, remaining on this theme of the anti-vaccination movements, which have very much defined the history of vaccines, even as vaccines have, ultimately eradicated smallpox, for instance. We also have the history of this kind of recurring concern. We might call it vaccine hesitancy, but also vaccine animosity over the course of the history. Here are some images from the 19th and early 20th century capturing some of these concerns. Diane or Paul?
Sure. I think I think at the heart of this, I mean, obviously the birth of the first anti-vaccine movement was with the first vaccine, with the sort of anti-vaccination legs following the smallpox vaccine. But I think at the heart of this is the notion of being compelled to do this, that you are compelled to get a vaccine. I think that's really where the pushback comes. Even if you ask modern anti-vaccine activists, what is it that you want? They'll all say the same thing. I want vaccines to be optional. I don't want to be told I have to get them. I think that's, and so in order to do that, in order to sort of push people away from mandates, then they create these images, and many of which obviously are based on false notions, but I think that's the heart of this.
Yeah, and
I would also just
so go ahead. Go ahead.
Sorry. I was just going to mention also, the idea of how people look at risk. And, one of the things with vaccines is we do give vaccines to otherwise healthy people. It's a preventative measure, and that always brings a different kind of risk into the picture.
Mm-hmm. Yeah, I think one of the things I would add to Paul and Diane's point is that there's a history of the question of how you balance individual liberty, the right to refuse, versus the policing of the public health. And this image really captures this history that we often don't think about in public health, which is the police function of public health, to kind of mandate or to really drive the interest of the broader public, even if it means overriding an individual's concern.
I think it also that the way vaccines, and here this is a good lead-in for this, the way vaccination is done and vaccines and how pure vaccines are was very, very different from what we see today in the manufacture of vaccines. And here we see some of the devices and the vaccines that were used through the 19th century. And it was a period of a lot of experimentation, a lot of debate about the proper methods of vaccination and what were the most effective methods of vaccination, as well as what were the most effective ways to preserve the vaccine itself. And vaccine was generally done, for the most part, what we call arm to arm. And that is that the viral vaccine material was taken directly from the blister or pox or vesicle that formed on a person who had been recently vaccinated, which in itself, even today, people kind of say, "Ooh, what was going on there?" But it was taken, straight from that sort of blister, if you will, and then inoculated. The next person was inoculated. And another way of doing it was through using the scab that formed on a vaccine. And those scabs could be preserved for a while and were even sent through the mail. In fact, this wonderful little device, which isn't much bigger around than a half dollar, and looks almost like a little cosmetic balm or something, was a case for preserving a scab, or what was called a crust, from a vaccine. And it was used by a Baltimore doctor through the mid-19th century. In fact, is engraved with his name. And I think just looking at the material used here and the workmanship of this little case speaks to how kind of precious this material was, this vaccine material at that time. The device up on,
go ahead.
Go ahead.
No, go ahead.
Ask a question. I was just going to explain the Vaccinator device. There were many different vaccination devices developed and patented in this sort of fairly free market. This device was designed, that little tip on the end that is protruding there was hollow at the end and could hold a little bit of vaccine material. And you could cock this device, it was spring driven, hold it against the arm of the person being vaccinated, and then release the spring. And that little protrusion would sort of cut into the skin and actually deposit a little bit of vaccine under the skin. And then another thing we see here are small little capillary tubes that are filled with the vaccine, the viral material. Actually, this was probably the most popular way, the way it was manufactured by the end of the 19th century. And indeed, it was coming straight from cows. Cows were being inoculated, right, and no longer arm to arm.
Right. So a whole world of technology that we have. We think of vaccines and we think of tiny syringes and needles, but there's a whole complex technological history to the world of inoculation. So this brings us to the late 19th, early 20th century and the bacteriological revolution, but also another world of implements like this that really symbolized the vaccine effort and the effort to win the public over.
Mm-hmm. We start seeing here also the sort of the advertising that's going on. Certainly the pharmaceutical industry is getting much more involved in the manufacture of vaccines at this time. And Mulford is one of the companies who's very much involved. And they indeed develop this, what's a vaccine shield, something to go over, the sort of the vaccination spot, the area while it heals. But also this wonderful button, this sort of, I have been vaccinated, this sort of pronunciation that, I am protected, I have been vaccinated, which is both sort of a personal statement, but speaks to that vaccination is this kind of communal activity. It is sort of a political activity, and here also a commercial activity.
Great. So this then brings us to the era in the late 19th and really early 20th century, which is associated with several milestones. So where at first you have things like vaccination and smallpox, the term vaccination becomes much more widely applied to a wide range of other bacterial caused diseases, anthrax, rabies, diphtheria. And I wondered if we could say a little bit, John, about the emergence, these milestones, these waves of animal-oriented vaccines, starting with anthrax.
So just reflect that from Jenner and smallpox in 1796, it was 90 years until Louis Pasteur got around to, or had the thought to prevent rabies by vaccination. But just a few years before that, 5 years before that, Pasteur was working on animal diseases, silkworm famously, but anthrax of livestock, of sheep and of cattle. And so his first bacterial vaccine was a anthrax vaccine for animals. So this is a reminder that we ignore animal health at our peril and the economic and commercial value of livestock. So he develops an anthrax vaccine in a controlled experiment. He vaccinated 24 sheep. He left 24 control sheep, came back a month later, gave them a lethal challenge. All but one of the vaccinated sheep lived. 100% of the unvaccinated sheep died. And so what you're looking at here on the left is a veterinary preparation of anthrax vaccine. Of course, in our modern time, we now think of that as a bioweapon threat, but it was and it is still an economic threat from the livestock perspective. Diane, I'm going to ask you to help me with the Sheridan's Cavalry condition powder, because I think this was an additive to feed, but I'm going to let you help me out on this one.
Yeah, it's an additive to feed, and somewhat like the patent medicines that we're familiar with from the 19th century that were manufactured for people. There were many similar medications manufactured for animals, veterinary medications that indeed claim to cure just about everything. But I think it's a little hard to read here, but you can see that it says actually on this condition powder that it's a preventative and a cure for cattle plague, including anthrax, as well as rinderpest and some other diseases. So I think I'll echo what you said, John, about the economic importance of animals, and that many of these medicines, the work done in veterinary medicine in America, there was the Bureau of Animal Industry, did really important work and research that also led in the future to human vaccines for humans too. So there's a lot of crossover there. Obviously, at this time, One Medicine was very much talked about, and we talk about One Health today, and of course it continues to be true.
Yeah, I think that we'll move on to the next slide on rabies, but it also illustrates this notion that, that animal health and safeguarding the health of animals because of their commercial and agricultural importance was very much a leading edge of vaccine development in this time period. And then later, human health, and that the sort of the politics, therefore, of using vaccines being shaped by the quest to protect livestock is an underappreciated aspect of the history. And so that brings us to rabies, which is not protecting dogs as food, but dogs as, domestic pets. John or Diane, any observations on the first rabies vaccine, which is also developed by Pasteur in the 1880s during this heyday of bacteriology?
So the kit shows that it was a regimen of 21 injections, and Pasteur's original was that they would increase in potency from the first to the 21st. I think by the 1920s, as you're seeing here, I believe each of those vials is equally potent, and the understanding of what it took to induce immunity was a little clearer by then, but still, nothing like the molecular biology tools we have today. On the rights, the veterinary rabies vaccine, obviously, and we're familiar with getting our companion animals protected, which we do for their own sake, but for our own safety as well.
Right. And so moving on to another development in, you might say, the use of bacteriology, which is the backdrop here, the discovery that there are microorganisms that can be visually identified through microscopes that are causative agents in many of these ailments, diphtheria, rabies, anthrax, tuberculosis, which doesn't yield to a vaccine, is very much part of the backdrop of the story of vaccines in this time period, and which brings us to the development of the diphtheria, not vaccine, but antitoxin, which is a very different way of biological defense against a bacterial ailment. Diane?
Yeah, I guess first I'd just like to say that, the impact of this particular product, the antitoxin in the 1890s, was huge. And I think it's always good to sort of step back and try and think about a time when diphtheria was a major killer of children. I mean, it was truly a much feared disease. And we're fortunate not to think of childhood mortality in the same terms. And the antitoxin itself was sometimes referred to also as sort of a passive immunity. And basically people are borrowing, if you will, the immunity from another animal. And in this case, usually the horses were used. And I think our next slide will show us a little bit more about the process for producing this particular product, which was used more as a treatment, though it did confer short-term immunity. And basically a horse or horses were immunized against the diphtheria. Their bodies built up immunity, thus, or as you might think of it, antibodies in their blood. They were then bled, and the serum was used to produce the antitoxin, again, used to treat diphtheria, but also to confer short-term immunity for diphtheria. And it was really groundbreaking at its time. And here we can see on the one side, we have the photograph. It's the New York Public Health Department, and they're producing the antitoxin. And here we see them actually bleeding a horse that has been immunized against diphtheria. And in the center there, we actually have the animal record book where they kept careful notes about both in immunizing the horses they were using and then bleeding them and using that serum. In fact, there's interesting little note here about some of the 460, some of the material that was actually discarded because I think it says it was moldy.
Right.
But interesting, just one last point here is that the health departments were major producers at this time of this material.
Yeah.
The note at it is that the pharmaceutical companies also made this, but they made toxin, but it was also made by health departments, including a health department in St. Louis, which when they harvested, they would inoculate the animal, in this case, a horse named Jim, with diphtheria toxin, and then they would harvest antitoxin from the animal. The animal actually had been infected with tetanus and ultimately was put down. And then what happened was they actually mixed the serum, because they did actually a very sensitive guinea pig test to make sure that there wasn't tetanus in those sera. Tetanus was relatively common in animals. And so, but the sera, the animal who had gotten sick and died, it was in August, they had took the September antisera and actually ended up mixing the two. And that led to 13 deaths in St. Louis and ultimately the Biologic Control Act. It was, there's an old line from a historian named Michael Harris, which is the history of drug regulation in the United States is built on tombstones. And that was true.
Yeah, I mean, you're highlighting a couple of things. One is that we sometimes forget the severity of these ailments that are the focal point for vaccine or antitoxin development. The way diphtheria would create this film around the throat and gradually asphyxiate children so that they would cease to breathe, and how quickly the antitoxin could actually counteract the toxins that were produced by the bacteria. But secondly, how the world of vaccine development also created these different pathways for producing the biologicals that then needed to be regulated, right? Because we had to be sure or more certain of the safety of the antitoxin as it's being developed by way of livestock, and to track it. So this then, we have about two questions that have popped up that I thought I would throw at you before we move on to the next slide from the audience. One, I'll just throw them all out and see what you have to say. One is that, so in the past, have vaccines caused ill or detrimental side effects? That was one. And I think we just talked about one of them in Paul's case. Do you have examples of, quote, quack vaccines that promised cures? And then third, did the canine inoculation against rabies come later after the implementation of rabies vaccination for humans in Europe and the United States? Any observations on any of those?
So yes, to amplify Paul's comments, there have been manufacturing mistakes of a variety of times, and in an episode caused some deaths. And each time, the regulation of what we now know as the FDA became more and more and more stringent. And so and we could take an hour and talk about the progression of regulation of the industry and the number of safeguards. That's really remarkable as well.
Mm-hmm.
And there certainly were quack vaccines. I mean, they were called bacterins. They were really developed before the FDA in 27. And you would just take a bacteria like E. coli or Klebsiella or Proteus or group A beta hemolytic strep, grow it up, kill it with carbolic acid, phenol, and then call it a vaccine. I mean, they were cheap, they were easily made, they were very lucrative for these companies, and they didn't work.
Mm-hmm.
But they didn't have to work. They didn't have to be shown to work. There was
no requirement.
Yeah.
And then Pasteur was using rabies for post-bite prophylaxis at first. And so yes, essentially it was human treatment, and then prophylaxis came later.
Mm-hmm. In the US, really the laws, really about the mid 20th century is when we really became serious about vaccinating our pets. And most states did, come up with laws to that effect. So that's what we saw in the previous slide. And as far as other kind of side effects or things, I would say with the serums, one of the first things that showed up also was something that became called serum sickness. And we have another slide. I think we might touch on this a little bit, but again, we're talking about the immune system and vaccines, and they do promote an immune response within our bodies. That's how they work. But there can also be some substances in the vaccines that promote an allergic response in some people. And certainly with these early serums that were produced from horses, there could be some proteins, from the horse or the antibody from the horse that would provoke some allergic responses in people.
Right. So one of the fascinating stories that are that's playing out today, but has been historically true, is getting vaccines to their destination is a continuing challenge. And one of them is captured, one of those moments is captured in this slide, the famous challenge of getting the serum, the diphtheria serum, to Nome, Alaska in 1925, also known as the Great Race of Mercy, or the Serum Run, that was involved a dog sled relay across the US territory of Alaska by 20 mushers and about 150 sled dogs, which made the name Balto, one of those lead dogs, quite famous. And here you have a statue of Balto standing in Central Park. There's if not Disney, there's certainly a cartoon movie about Balto that came out about 20 years ago as well. Any references? I mean, this is part of a broader issue that we're grappling with today, which is, how do you get the vaccine to its destination? So, John or Paul?
It was obviously harder. We don't have dog sleds to deal with today, but it's still temperature control. There, the middle picture says it was 1,000 units of biological activity in that syringe. It's changed color over time. It would have been straw color when it was fresh, but by the time it got there, maybe it was delivering 800 units of activity. And so what we've learned over the century in temperature, and you've read the news articles about the need for ultra freezing with the modern COVID vaccines, and so we've learned as we've gone along.
Mm-hmm.
Yeah, Kevin Bacon, by the way, was the voice of Balto in that trial.
Kevin Bacon, he's everywhere. I didn't realize that.
Well, we've connected to him.
Yeah. So, of course, that brings us to another instance of vaccine, the tetanus vaccine, which takes us through the decades in the interwar period, 1920s through the 1930s, 1940s. John, could you say a little bit about this bacteria that lives in soil and dust and manure, and the kind of produces painful tightening of the muscles, among many other symptoms, and the quest for a vaccine?
So that's tetanus, what you just described, and it causes contracture of the muscles. It's in the soil. It has the reputation of being the rusty nail disease, but nowadays, soil that you have brought into your house, an indoor wound, is actually a more common cause of tetanus than an outdoor wound. Speaks to how much we spend time indoors. So World War I, a lot of casualties in Europe, a lot of skin breaks, soil introduced into the skin. Tetanus is a major problem. Tetanus antitoxin, analogous to the diphtheria antitoxin, somebody else's antibodies, is used as a treatment. Scientists eventually in the late 20s, early 30s, developed tetanus toxoid, a modified toxin that induces immunity instead of inducing disease, and it can be used for prophylaxis. And that's the major advance. We then go into World War II. And so what you see here on the pictures are identification tags, identification discs. On the left side is a sailor's tag. The Navy would call them discs for obvious reasons. But if you look at the fifth line, you'll see T7/42 and then type A. So he got a dose of tetanus toxoid in July of 1942. So right there on his, what we call dog tags, was part of his vaccination record in metal, type A being his blood type. The one on the right is my father's. He was a lieutenant in the Army Air Corps, and you see the second line is his serial number, T4243. He got 2 doses. O is his blood type. I was surprised that the RH factor, positive, negative, is not on either of those tags. That was just coming in from the blood banking perspective, coming into an understanding at the time. But here you see metal immunization records.
Great. So I'm gonna do 2 slides, skip past one, which is just to point out that as vaccines become more widely used, other issues grow. And we've talked about the production process, which needs oversight, regulation, but also the fact that allergic reactions also need to be attended to by the rise of the use of medic alert bracelets, especially to signal allergies to horse serum. So as vaccines develop, there is also this other kind of management administrative process that's necessary. And the other thing I want to point out is that before we discuss influenza vaccines, it's important to point out that many ailments were just not amenable to either bacterial analysis because they were caused by viruses, sort of submicroscopic organisms in the early years, but also impossible viruses, impossible to isolate, let alone to study and to produce vaccines. And so the influenza vaccine is an interesting story, which isn't part of that early era of bacteriology. And yet there were purported influenza vaccines. John, Paul, would you like to comment on the influenza vaccine story and where it fits in this timeline?
John, you can see this. You can do it because you were alive in 1936.
Almost, Paul. So the swine flu pandemic of 1918, there was no ability to visualize viruses, and so they would take cultures out of the lung and they would be able to grow bacteria, and so they blamed the bacteria they found on the pandemic, even though it was a viral pandemic, not a bacterial pandemic. Nonetheless, people made some bacterial influenza vaccines. You see a obsolete version over on the left. It was actually a mixture of 6 different kinds of bacteria. And then over on the right is a modern day P-Vax-Hib, the true influenza bacillus, Haemophilus influenza type B, of modern vaccine to prevent a major cause of meningitis in children at the time, and now that disease has almost entirely disappeared. In the middle is one of the early viral vaccines. This one manufactured by Squibb, and you can see that it contains 2 influenza virus types, type A and type B. It's a combination vaccine. Paul, what do you want to add?
No, so I think there's a mumps one coming up next, which I have a story that will be coming.
So,
no.
Yeah, the mumps, we are a couple steps from mumps. But let me just skip ahead because this does take us to the advent of these combination vaccines. That is to say, there's this interesting selling line here from E.R. Squibb that says, don't use your young patient as a pincushion rather than giving them lots of different vaccines. How about combining them into one?
Convenience is a major factor to, as the number of diseases that are preventable rises, how can we avoid inconveniencing the patient, the parent? And so can we give instead of 3 jabs, can we give 1?
Not so easily done, by the way. I mean, often there's competing stabilizing agents, buffering agents. It's not easy to put 3 different vaccines into a vial, especially for the live attenuated viral vaccine. So it's easier said than done.
Right, and so we have a couple of questions that have come in, and they run the risk of sending us back in time. One is, did the Pure Food and Drug Act of 1906 address vaccines? The second is, let's see.
No, I'll give you the answer. No.
Right, there we go.
The Biologic Act predated the Pure Food and Drug Act. It was 1902.
Yes. And then could Diane tell us a bit about what is involved in maintaining these vaccine specimens in museum collections?
Sure. First of all, I will say that we are not maintaining vaccine specimens for them to be effective in the years to come. So we are not keeping them at especially cold temperatures. So that's not really why we're saving them. So I will mention that first off. Other than that, I mean, honestly, as we've seen and as we hear all the time with the COVID, that really the effectiveness of these vaccines is going to be destroyed pretty quickly by just keeping them at room temperature. So they're not active vaccines in any way.
Right.
And they are kept pretty much like our other collections are kept. I mean, we keep them in locked cabinets. We try and control temperature and humidity as much as possible. We keep them in, secure storage areas that we, again, it's really environmental controls that we focus on the most.
So we have about 4 slides coming up. We're running a little behind time, which really turn our attention to polio, which for the people living in the 20th century has loomed large in the story of childhood disease, less associated with, well, less mortality than paralysis, but certainly also mortality, but also in which the story of the vaccine figures prominently. Waves of epidemics that cause paralysis associated with summers that create anxiety, leading ultimately to the creation of vaccines in the early 1950s. Any observations about these artifacts from the polio era that predate the vaccine?
I would say this one thing just about the vaccine, since I was actually in a polio ward when I was 5 years old for about 6 weeks. So this polio has always been a very emotional thing for me. But when Jonas Salk made his vaccine to prevent this horrible disease, I mean, he made it by taking the virus, growing it up, purifying it, and killing it with formaldehyde, and showed that in 700 people in the Pittsburgh area that it appeared to induce an immune response, which he imagined would be protective, and it was safe. Then there was the biggest trial, arguably, of a medical product ever performed. I mean, there were in that between 1954 and 1955, the so-called Francis Field trial. They inoculated 420,000 children with Salk's vaccine, 200,000 with placebo, and then another 1.2 million children served as observed uninoculated controls. That was a 1.8 million child study. And what they found was when Thomas Francis got up onto the podium at Rackham Hall to announce that this vaccine worked, he said the 3 famous words: safe, potent, and effective. And that was on the headline of every newspaper. The reason he knew it was effective was why it broke Jonas Salk's heart that this trial was ever done. The reason he knew it was effective was because 16 children in that trial died of polio, all of whom were in the placebo group. The reason he knew it was effective was because 36 children had paralysis or were in iron lungs. 34 were in the placebo group. That's how you knew it was effective. And I think that often gets lost in these trials. I mean, that when that case was made, those statistics, I guess, were numbing and distancing. But the fact of the matter is, children had to suffer and die in order to prove that point. And the point being, I guess, that the price of medical innovation is invariably comes with a human price. And that was certainly true here.
Mm-hmm. Mm-hmm. And there's also the story of the Sabin vaccine as well, the injectable versus the oral polio vaccine, which intersects with the Cold War. Vaccines have always had a kind of geopolitical dimension to them. And Paul, any quick comments about the Salk versus Sabin story?
Right. So the Salk vaccine came in 1955. It reduced the incidence of polio from about 15,000 cases a year to about 900 by the time that the Sabin vaccine was introduced, which is the oral polio vaccine. And then that eventually eliminated polio from the United States and ultimately by the 1970s, late 70s, and from the Western Hemisphere by 1991. But it too came with a price. I mean, the oral polio vaccine was itself a very rare cause of polio. It occurred in maybe 1 per 2.4 million doses. It was rare but real. And for that reason, in 1999-2000, we moved back to the Salk vaccine because we were unwilling to tolerate the 8 to 10 children every year who got polio from the polio vaccine when we had a vaccine like the inactivated vaccine, which was also safe and effective. And a number of countries, including Scandinavian countries, were able to eliminate polio from their country without ever going to the Sabin vaccine.
Mm-hmm. So that also highlights the kind of fits and starts that are very much part of the decades, sometimes century-long challenge of disease eradication. Ultimately, stories like smallpox, heading perhaps that way towards polio on a global scale. And then there's this dimension of the polio vaccine trials, the need to actually administer, track, understand, and study. And I think, Paul, you just alluded to some of this, right? The use of early computation, punch cards, which have the region, state, and the county, the child's age, the school, the date of vaccination, and some other important data in order to do the work of actually determining the success or failure of a trial.
Yeah, impressive. That 1.8 million child study would have probably cost $6 billion today.
Huh.
It lived on a lot of volunteers.
So a couple of questions about polio. Does anyone know, sorry, let me just make sure I'm getting, does anyone know what the mechanism was used to ensure that kids in the placebo arms got the effective vaccine later? That's one question. The second, does anyone how have the ethics regarding vaccination trials evolved over time?
As I recall, the vaccine later, whether they were in the placebo group or the other group. Is that correct? Is that, or it was...
I think the national vaccination program, to get all children vaccinated rolls out fairly quickly. Paul, you'll know this better than I. Within a year or two, right?
Yes. No. I mean, that was Warp Speed 1. I mean, we mass produced, these 5 companies mass produced vaccine at risk, not knowing whether the vaccine was safe or worked. But when it was announced in, I think, April 12th, 1955, that vaccine worked, it was then licensed essentially in about 2 hours. It takes a little longer to get a license today, but it was licensed in about 2 hours, and it rolled out right then, immediately.
Yeah.
Yep, great. And I think the other thing that we'll want to just discuss briefly is, once again, the organizational and administrative challenge of carrying off a mass polio vaccination program. I mean, arguably, we had many, many years as a society to organize ourselves for that undertaking. As a childhood disease, it figured very prominently. And also, as some of you have written about the fact that, the polio vaccine trial comes in the wake of other large-scale developments like the Manhattan Project for building the bomb, or the production of penicillin, scaling up for wide use. And so American society in 1950, there's this incredible faith in the ability of science to be organized between private and public sectors to actually protect the public health and deliver on vaccines in the name of safeguarding the health of our children.
And these are mass vaccination campaigns, which is exactly what we need now. But we saw polio as a shared national tragedy. It pulled us all together. And maybe it's because we were coming off the shared national tragedy of World War II. It was certainly a different time, but it's harder to watch what goes on today, where it feels like we're not getting together. Rather, it's just more finger pointing than pulling ourselves together.
Yeah, the one quick thing I would like to say about the ethics is that, just like, as Paul said, there are these sort of undersides to the polio, the Salk vaccine. People had to die. There are also, what we might look back and see as really deeply worrisome ethical problems. The use of children, which is the norm at this time, the use of mentally ill children in a home for what are called mentally retarded back in the early 1950s for the trials. Or it's not an era that has the same kinds of safeguards on issues like, informed consent, or the right to refuse, or protection of vulnerable subjects in the service of medical research and knowledge development. So it's important to kind of not have an entirely rosy picture of the processes that produced these kinds of developments. So this will take us into our last 2 examples, and then we'll sort of end with, well, maybe 3 or 4 examples. We have mumps, rubella. I'll just put these up in sequence: rubella and measles, which are often lumped today as MMR, right? But they're very interesting stories turning to these childhood campaigns of the 1960s and 1970s that really become sort of follow on the wake of the polio vaccines and become the new face of what we expect out of vaccines. John, Paul, Diane, any observations on any of these kinds of childhood mass vaccine programs of the 1960s and 70s? And these,
if I could just take, if you go back a couple slides to the mumps slide, this is just a sort of an interesting story in the sense that The girl you see on the left is Gerald Lynn Hilleman. Her father, Maurice Hilleman, arguably is the father of modern vaccines. He's did the primary research for development on about 9 of the 14 vaccines that we currently give to children today. She got sick. When she was 5 years of age, she had the mumps, 5 years old. She goes into her father's bedroom, wakes him up and says, "Daddy, my neck hurts." He looks at her, sees she has mumps, and then does something no father ever does. "Wait right here," he says. He goes into the laboratory, with Merck actually, and he gets a swab, he gets broth, he comes back, he swabs the inside of his daughter's mouth. And then over a 4-year period between 1963 and 1967, he makes a live attenuated mumps vaccine. Then what he does, which you see here on the right, is Bob Weibel, who was a vaccine researcher, inoculating now Gerald's younger sister, Kirsten Hilleman, with a vaccine that ultimately protects her, which is interesting because normally older children bring viruses into the home and infect the younger children. Here, the older child gets mumps, the father makes a mumps vaccine and then uses that to protect the daughter. This is not the usual story, but.
Not the usual story indeed.
Great story. Yeah, I just have to comment on the rubella because the vaccination campaign was a little different, and partly because rubella isn't really, for the child who gets it, it's not really that dangerous a disease. I remember having German measles, rubella myself. But what they found was that if a woman who is pregnant gets rubella, then the fetus is very much at risk, either for, actual, all sorts of sort of lifelong disability. And so that was called congenital rubella syndrome. And so the campaigns for rubella, and what you see here, it's today's little people protect tomorrow's little people. And this really speaks to that idea of the community effort or communal responsibility, because they really had to reach out to people and say, your child, we need them to get vaccinated in order to protect these other people, the unborn child. And, I think that's another thing that really resonates today, is that vaccination isn't always just for the individual, the protection of the individual. It's very much a communal thing. And that really came out in the rubella campaigns. And you see it here, less of a menace. Why is Dennis less of a menace? Because he's not going to bring German measles to a pregnant woman.
So we have a question about about disparities at this point, which is we have several questions about inequality of access to vaccines and racial disparities in care. Does anyone want to? And it's interesting that this Stop Rubella campaign shows awareness of the need to picture kids, Black kids, white kids, and anyone want to comment on the way in which these campaigns are sensitive to or differential uptake of vaccines?
I'll take you to a federal law adopted in 1987 that produced a program called Vaccines for Children. And where there had been disparity in communities of color being under-vaccinated, it basically took the economics out of the way, and the federal government provides the vaccine. So that today there is no disparity in children on the basis of ethnicity or race. And it's a major success.
Mm-hmm.
And certainly those rubella vaccines and those campaigns you see, they're almost always, it's free vaccination. So, and they're big, they have vaccine days, a big communal effort, all out to get children vaccinated.
So then we'll sort of head towards conclusion. We'll run over by about 5 or 6 minutes or so. We have two stories really of the 70s into 1980 that are worth talking about together. We have the story of a prominent example of the swine flu vaccine from 1976 and how vaccine and its unintended side effects can cause lasting damage to the vaccine campaign. But then we also have the story in 1980 of smallpox eradication around the world, and the way in which 4 years later you have the decades long, the culmination of a decades long effort to rid the world, not just the country or not just a neighborhood or a region, but the world of smallpox, which is where we started. Any comments from maybe John on the swine flu story, or Paul about, and Diane about smallpox eradication?
So 1976, there was a few cases of unusual looking influenza. The government mobilized a mass vaccination program. That's the photo that you see there. It turned out that the disease did not spread. And so there was no epidemic, true epidemic, but there was the mass vaccination program. And it's a lesson for today. There was great concern that the vaccine caused heart attacks, and that was not true upon further review. But eventually there was a recognition that the vaccine, in about 1 in 500,000 cases, can cause a paralysis syndrome called Guillain-Barré syndrome. And so 1 in 500,000 people had a bad side effect, very, very bad side effect from the vaccine. And there was no disease to make that side effect, quote unquote, worth it. So the program stopped prematurely.
Right. And there was also a story of this sort of this rapid scaling up of the vaccine in the context of an election year. It has some resonances with what we've seen this year as well. And then you have this dramatic story of, which is decades in the making, of the eradication of smallpox. Which maybe no more needs to be said about that, but Diane?
Well, I mean, it's really an amazing story to eradicate a disease. Of course, we've had scares of smallpox as a bioweapon since that time, but in 1980, it was declared as, globally eradicated. This disease that had really, haunted mankind for who knows, as long as we knew. And, again, it's the effort, it's how do you do that? The administration, the distribution, how do you get that the immunizations to the people who need them? And, some of them were administrative and some things were technological that helped. And the bifurcated needle down there at the bottom as a vaccination device. And I think, earlier on, I showed some of these early 19th century vaccination devices. Well, this simple device was easy to use. It didn't waste vaccine. It could be sterilized and reused. Again, people in the field could be taught to use it. And, just a small technology like that did make a big difference in this.
And it was better than that big complicated thing you see over on the right.
Yes.
So one question quickly is, why the bifurcated needle?
The bifurcation held just a very small quantity of liquid, and then it poked just deep enough in the skin. It was perfect.
Yeah. And with smallpox, you really don't go deep in at all. It's not like we think of a shot or a jab today, really, so much as only just sort of scratching the surface almost. So.
Right. So in some ways, technologically, we are back to where we began.
It doesn't look that different in some ways than some of the devices that were designed to hold a little vaccine and just insert it in the skin a little.
Yeah. I think the reason we were successful
Other things changed a lot.
I think one thing that made this easier was that smallpox really doesn't have asymptomatic infection. If you're infected, it's evident. And so it's easier then to do this so-called ring vaccination where you could inoculate those who were in contact with someone who was infected and those around them and those around them. As distinct from, say, the SARS-CoV-2, which has a large sort of base under the tip of the iceberg of asymptomatic.
It doesn't have an animal reservoir either. There's other things that make a disease,
interesting,
hateable.
Yeah. That's an important point. So let's wrap up by talking a little bit about vaccines unrealized. I mean, while we want to highlight the disorders for which there have been effective vaccines and the complexity of how they've been deployed and what difference they've made in the history of the nation and the world, there are also examples of unrealized vaccines. hopes for a vaccine to HIV/AIDS, for instance. And then there are new vaccines that have come by way of new biological understanding, like the emergence of recombinant DNA vaccines. And then our last slide will turn to COVID-19, but I thought maybe I would ask Paul, Diane, or Paul to weigh in on the story of HIV vaccine and the hepatitis B vaccine.
I guess, when HIV emerged in 1981, I mean, what had just happened? You were just talking about smallpox eradication, the development of quite a few successful viral vaccines in the decades before. Here we are in the early 80s with a pretty positive attitude, if you will, about vaccination. And there were definitely promises early on that we would, have a vaccine in a couple of years. And of course, the virus, we didn't really know that much about the virus. And I'm sure Paul or John can speak about, some of the reasons why a vaccine has been difficult to produce. But, those promises were not fulfilled.
Mm-hmm.
The hard part about it, I mean, influenza is hard because it mutates from one year to the next. Frankly, it mutates almost from 1 minute to the next. It's constantly drifting. AIDS or HIV really mutates during a single infection. I mean, you make antibodies to the original virus that neutralize the virus, but the virus keeps mutating, plus it infects T cells, which are sort of the orchestrator of the immune system. So it's constantly mutating while it's sort of paralyzing your immune system. That's a bad combination. It's mm-hmm. not for want of money or want of effort for an HIV vaccine. It's just been that hard to make.
Mm-hmm. And as I understand also, people don't really get HIV and recover, so that the whole sort of the immune response is very different. If you think about when we get, people know you get measles or you got smallpox, if you did live through it, your body had an immune response that protected you. And I guess right. HIV, that's also a different way that particular virus operates.
So perhaps in the interest of time, we'll end with this perspective slide to show where we've been, not just in this conversation, but historically, and ask, the speed with which the COVID-19 vaccines have been produced is notable. The challenges of distributing them to the population is not unique. And I wondered if you could say a little bit, with history as your backdrop, as to where we are today and the likely impact of these vaccines, not only on COVID-19, but perhaps on vaccinations more generally. That is to say, how are we likely to emerge out of this? Where are we in the chapter of vaccination as we confront COVID-19?
So this graph shows the accumulation of diseases of humans that are preventable. You see that big gap between smallpox and rabies I mentioned earlier, and then the accumulation accelerates. And the biological sciences just get better and better, from the 1930s onward, the ability to grow viruses, the ability to isolate toxins and understand at a molecular level what's happening. And so with each advance and each technological improvement, we science can get better at efficacy, can get better at safety, can get better at manufacturing quality. But those bugs, those pathogens, those viruses and bacteria are still elusive and give up their secrets begrudgingly. And I think we've found out that the virus that causes COVID-19 is to a certain extent, an easy virus from a vaccine production standpoint. So we lucked out, but we also have the benefit of tools that didn't exist even 10 years ago to apply to the task.
Great.
And then there's the sociologic pieces. We talked about mandates. We talked about equity. We talked about disparities. So go back to that first satire cartoon that I called the Internet of 1802. The human nature piece of this is even more daunting than the microbiology pieces of this.
Mm-hmm. Diane or Paul, any closing comments? We have a question here, which is, do we know anything about the campaign to distribute pins, stickers, public media campaign about the COVID-19 vaccine?
I have not seen any, but I'm hoping that anyone out there who gets back, or when you get vaccinated, that you hold on to those, those little items. And I'm sure there got to be some buttons or stickers. They usually are now, but the only I've seen are the little vaccination cards, the CDC printed cards that has like the date of your first. When you
so a bunch of hospitals or health systems have made their own buttons, and I've seen a template of the CDC.
Oh, so. Okay. Well,
one of the things, Paul, did you want to jump in?
No.
Oh, okay. I was going to say one of the things that challenges us with COVID-19 is just simply that we have a lot of historical examples, but nothing that is in any way comparable in terms of the scale of this pandemic in our lifetimes. We'd have to find somebody who's over 100 years old, who was sort of sentient and aware in 1918 or 1919 to describe this. And so there's a sense in which maybe the scale of this is enough of an incentive that's doing the public health work all by itself. I mean, there's the number of deaths, the scale of the illness. And frankly, you know, it's taken us just about, 8 or 9 short months to develop a vaccine. You know, it'll take us a while to figure out a public health campaign, to get everyone vaccinated. And we've been dealing with mostly misinformation about the vaccine and about the public health challenge. So we're opening a new chapter really in January 2021 that may in fact see something that looks like buttons, pins, and campaigns to win over the public. Mm-hmm. Well, with that in mind, Paul, were you going to jump in?
No. Again, I'm just.
Oh, okay. I'm reading body language, which is always dangerous via Zoom. So I know, Diane, that you want to say a quick word about the Smithsonian American History Museum exhibition.
I just want to mention for everyone that we are, despite the fact being at home, like many people, we have been working on an exhibit started before the pandemic, actually, on sort of the history of medicine. It's called In Sickness and in Health. And we hope to be opening in 2022, and that will see many people back at the museum.
Great. So I want to close by thanking Diane, John, and Paul for what is, in fact, just a marvelous conversation, but also a whirlwind history through through vaccination, inoculation, variolation, that really properly gives us some insight into the challenges that we face today. And I'm a strong believer in the fact that if we understand not just the victories that are created by vaccines, but also the challenges and in many ways some of the problems, we'll be much better equipped to manage our way through COVID-19 as we look ahead, hopefully, into 2021. So thanks again for this terrific conversation.